Synthetic chemistry has been one of the driving forces of the 20th century. Just if you think how chemistry has changed our lives, think about the different textiles we were able to synthesize with synthetic chemistry. Think about different drugs, antibiotics we have been able to synthesize with synthetic chemistry. And think about different materials, for instance, like plastics. So synthetic chemistry has changed our life and has been one of the sciences of the 20th century. But I also think that synthetic biology will become the science of the 21st century. If you think for a moment about biological systems, they have a couple of advantages over chemical synthesis. And one thing is that biological systems are, for instance, self-optimizing, which means that they really can improve themselves while they're operating. And that's a concept we would not find in chemistry. Biological systems are self-repairing. Think about catalysts that would be able to repair themselves once they are broke. And biological systems are self-propagating. Again, this is a feature you would not find in classical chemical systems. So there are a couple of advantages biological systems bring with them. And on top of it, biological systems operate in a sustainable fashion. And biological systems are also environmentally friendly. So one of the tasks and challenges of synthetic biology is now to really use biological systems to do chemistry, to synthesize new compounds and new products. And today I'm going to show you how we actually can come and reprogram the chemistry of living cells. And we want to do this with a concept that we would call synthetic metabolism. So what is the concept of synthetic metabolism? So the idea would be that we start off by designing theoretical pathways basically on the paper or in silico. Then we would go ahead, find and engineer the individual building blocks, the enzymes you would need to build these pathways. We would enter a cycle where we build and optimize the individual synthetic pathways. And finally, we would like to transplant these pathways into cells that would be mini factories that could produce products for our everyday life. So the central question of today's lectures will be, how do we design such synthetic metabolic networks from scratch? So what are the common design principles we have to use if we want to design metabolism from scratch? The second question would be, how can we realize synthetic metabolic networks in the lab? So how can we come from the in silico, from the design part, to the actual building part? And finally, the last question is, how can we optimize synthetic metabolic networks once we've built them in the lab? So basically improving these systems in the next step. So I'm going to exemplify these questions by talking about research in our lab in which we have tried to establish a process that we will call synthetic CO2 fixation. So what is synthetic CO2 fixation, and why are we interested in this process? Well, the answer lies in the air surrounding us. And as you all know, CO2 is a very potent greenhouse gas. I think we also agree on the fact that the amount of CO2 in the atmosphere has been steadily increasing over the last years, decades, even centuries. And of course, this has severe consequences. Just think about the phenomenon of global warming. So it's a typical view on looking at CO2 as being a challenge and a threat for science and society. At the same time, we can also look at CO2 being a chance. And being a chance because in principle, it's a very simple and ubiquitous carbon source that sits out there in the atmosphere. And we as scientists, we as a society, should be able to harvest this molecule and convert it into useful compounds. Yet, we cannot do that. So in fact, there is no chemical process, no catalyst, no simple chemical process in chemistry that would allow us to harvest atmospheric CO2 and to convert this CO2 in a sustainable fashion into a given multi-carbon compound. And all our chemical efforts are still outcompeted by biology that fixes carbon dioxide on a gigaton scale per year. And I think we all know very well the process of photosynthesis that takes, for instance, place in plants, cyanobacteria or algae. Yet, biology is not perfect. And it's not perfect because the systems have evolved over time and they were not designed a priori. And one problem, for instance, is that these systems are not running under optimal conditions, so they're not necessarily very fast. And another problem is that, for instance, the product of biological CO2 fixation is biomass. And biomass is not necessarily something that you can directly use in the chemical industry. And this has inspired us now to really rethink biological CO2 fixation. And we've come with the question, so what if you were actually able to find or design new CO2 fixation pathways? Pathways that would allow us, for instance, to convert CO2 directly into interesting product, or pathways that would operate faster than naturally evolved pathways. And this is then what we would consider a name synthetic CO2 fixation. So apparently one critical step in fixing carbon dioxide is to identify enzymes that are fixing carbon dioxide efficiently. So the very first step in this project has been to identify enzymes that could fix CO2 very efficiently. This slide reminds you of defining the efficiency of enzymes. So you want to actually have enzymes that have a very high kcat number, a very high turnover number, which means that they can convert a given substrate molecule at a high speed. So that's one part. The second part is the enzymes should actually have a very low Michaelis constant, so-called Km value. This would tell you that these enzymes can actually also operate at very low substrate concentrations. And what you want to have at the end is an enzyme that is operating at a high turnover rate at very low substrate concentrations. And this is what we would then call a catalytically efficient enzyme, so kcat divided by Km. And think about a moment about CO2 fixation. So carbon dioxide is very low concentrate in the atmosphere, only 0.04%. And you want to have enzymes that can fix carbon dioxide very efficiently at these low concentrations. So one of the first in this project has basically been to identify enzymes that would fix carbon dioxide in an efficient manner under atmospheric concentrations. And what we've done is in the first step is going through biological databases and identify the catalytic efficiency of different enzyme classes. And what we see here is basically different enzyme classes plotted according to the catalytic efficiency. And we're surprised to learn that the enzyme that is used in plants to fix carbon dioxide, Rubisco, is not the most efficient enzyme. In fact, there are other enzymes that can fix carbon dioxide more efficiently. And among these enzymes are so-called N-alkyl-CoA carboxylases, reductases, or short ECRs. And these enzymes are actually up to 4 times more efficient than Rubisco, the enzyme that is used in plant photosynthesis. So what are these enzymes? Well, the prime example is the so-called crotonyl-CoA carboxylase reductase. That basically takes a molecule of crotonyl-CoA, reduces this molecule of crotonyl-CoA with NADPH as cofactor, and introduces a CO2 molecule to give rise to the carboxylated product, ethylmalonyl-CoA. It's a very nice principle to be used, but it's only one reaction. And ideally, you want to have actually multiple reactions. You want to have a set of enzymes that can do very different carboxylation reactions. And one of our first efforts has actually been to open up and expand the biosynthetic space of this class of enzymes. So we've asked the question, can we now fit other substrates into the active site of this enzyme? Can we expand the substrate spectrum of these enzymes to catalyze other reactions just besides crotonyl-CoA? So we've addressed this in 2 different approaches. One way has been we've used enzyme engineering. So we've used crystal structures of the enzyme. We've looked at the active site pocket of these enzymes. We've tried to open the active site pockets so that other substrates could fit in. Another approach has been to use genome mining, which means that you go ahead and you just screen the biological diversity of this class of enzymes. In other words, you use the phylogenetic tree of these enzymes. Then you synthesize 30, 50, 100 different variants, and you simply test the substrate spectrum of these different enzymes. So with these 2 approaches, we were actually able to identify a set of enzymes that catalyzes very different carboxylation reactions. So we can now have enzymes that catalyze short substrates, and have enzymes that can carboxylate longer substrates, branched substrates, bulky substrates, different decorated substrates. In other words, we have now a set of enzymes at hand that can carboxylate with high efficiency very different reactions. So how can we now use these enzymes to build synthetic CO2 fixation pathways? How can we come from an individual enzyme reaction to a complete pathway? And this is now the second part of my talk, where I want to show you how we can design synthetic metabolic networks from first principles. So what we do in the lab is we simply start with a blank piece of paper and we start to draw artificial synthetic theoretical pathways. And all these pathways are very different, but all these pathways are centered on an individual ECR reaction. And even though these pathways have different topologies, and even though these pathways have different intermediates, they also share some common principles. And one principle is that all these pathways are actually cycles. What does it mean? Well, basically, the product of a carboxylation reaction is turned back to become the substrate of the carboxylation reaction. And this way, you can basically run in a continuous fashion carbon dioxide fixation, which means you can really fix carbon dioxide continuously. That's the principle of a metabolic cycle. So another common principle is that all these pathways are, again, based on an ECR reaction. And the third principle is that all these pathways have a carbon splitting reaction. For instance, shown here, you can take off some of the fixed carbon, and you can shuttle this carbon into an interesting technological compound or biological building block. Again, these are theoretical cycles. Don't forget, at this stage of the design, you don't have any specific enzymes in mind besides the ECR reaction. We just think about potential transformations. We just think about oxidations, potential oxidations. We think about potential reduction reactions. We think about potential CC splitting reactions. We think about potential hydrations or dehydration reactions. So these are all theoretical enzymes that might exist. So from this design, we actually also have to decide which of these cycles is now a really good cycle. So you need to evaluate the different cycles we have drawn. And so we've used simple chemical criteria to decide which of these cycles is actually the best cycle to be realized. And one of the important criteria is here, you need to have cycles that are kinetically favored. This means you want to have cycles where you actually have fast and efficient enzyme reactions involved. So all the individual reactions you would like to apply in your cycle should be fast and efficient, which would allow us to run the cycle in an efficient manner. A second criterion is actually a thermodynamic criterion. So all the cycles should be thermodynamically favored, which means you need to think how much energy per CO2 molecule fixed do you have to spend. And so we've done this calculation, for instance, for the catch cycle shown here. So the catch cycle can turn 2 times to produce 1 molecule of pyruvate. And while turning 2 times, it actually has to spend 4 ATPs for 1 pyruvate molecule fixed from CO2. Now you can compare this ATP efficiency with natural existing pathways. And you can actually see that the Calvin cycle, the pathway that is used by plants, bacteria, and algae in photosynthesis, uses 7 ATP per CO2. So in other words, the synthetic solution from the lab is almost twice as energy efficient in terms of ATP usage. So it tells you already that you actually can come up with solutions which are more ATP efficient than naturally evolved systems. And finally, the last criterion you want to actually apply is the criterion of thermodynamic feasibility. So to make sure that all the reactions you apply in your cycle have a favorable thermodynamic equilibrium. So in other words, the concentration and the equilibrium of the reaction should lie on the side of the product formation. In other words, the free energy profile across the reaction should be negative, and this will allow you to turn the cycle in a clockwise fashion efficiently without any major harms. So having identified and evaluated the cycles, we came up with the idea that the catch cycle that fulfills all these 3 criteria would be a very good starting point to build now synthetic CO2 fixation in the lab. So this brings us to the second part, where I want to tell you how we can now realize such synthetic metabolic networks from first principles. So on the one hand, we now have this theoretical cycle that we've drawn on the paper and that we've evaluated. On the other hand, we actually want to realize this cycle and build this cycle, which means we need to find all the individual biological parts. And a network, a metabolic network like the catch cycle, that features 50 different reactions, means that you need to find 15 different enzymes that could do all the different transformations. So what can you do? You basically can go back and you can go through different databases, like Brenda, KEGG, Stranda, or PubMed, to find enzymes that could do one or the other transformation. So right now we have more than 50 million annotated genes in databases, and we have more than 40,000 characterized enzymes in databases. So the chances are very high that you would find the enzyme reaction somewhere. Of course, you still need to express the enzyme, you need to purify the enzyme, and you need to test the enzyme. But ideally, you can find a lot of enzymes in these databases. Yet sometimes you find an enzyme in the database, but it does not have the properties it should have. And this is shown here. So we are looking for enzyme that could catalyze this reaction up here, which is a dehydrogenation reaction of a compound called 2-hydroxybutyryl-CoA. So in fact, there has been an enzyme that was deposited in the database. The problem is that this enzyme is highly oxygen sensitive, and it actually dies away within 15 or 20 minutes, lose all its activity. So how can you overcome this problem? How can you find a better way to do it? Well, one strategy is that you simply go ahead and you screen for biological diversity. So in other words, you try to express and test homologs of this enzyme. And so lo and behold, by using this strategy, you identified an homolog in a marine archaeon called Nitrosopromenes maritimus, which shares 50 or 60% of sequence identity, but was approximately 100 times more stable under oxy conditions. So looking for biological parts and screening different enzyme homologs can help you identify enzymes. But sometimes you have the problem that you would like to employ a certain enzyme reaction, but you actually don't have an enzyme in the database. And this is shown here, where you want to use a reaction in which a compound called methylsuccinyl-CoA is oxidized to a big-armed compound called mesocyl-CoA. Well, there's an enzyme described that can do this chemistry. The problem is that these enzymes are so-called dehydrogenases. And these dehydrogenases are very complicated enzymes because they oxidize the substrate, put the electrons onto a cofactor, and from this cofactor, the electrons are put in a second protein called electron transfer flavoprotein, and from there on, the electrons are put into membrane-bound respiratory chains. So in other words, if you want to use this kind of chemistry, you need not only the enzyme, but you also need this interaction partner, and you need a membrane-bound respiratory chain, which is awfully complicated. So in this case, it might be helpful if you simply shortcut this whole electron transfer relay. And what we intended to do is to convert this methylsuccinyl-CoA dehydrogenase into a methylsuccinyl-CoA oxidase, an enzyme that could directly use oxygen instead of having this complicated electron transfer chemistry. So what you need to do here is you need to look into the active site of the enzyme, and you should think about ways how to, for instance, accommodate an oxygen molecule there. So you can open up the active site by creating a bit more space. You can think about adopting and kind of allowing for a negatively charged to be developed, so putting more positively charged amino acids. And then you simply can go ahead and can test different active site mutations. And when we did this, we actually ended up and identified a triple mutant that could indeed catalyze the direct oxidation of methylsuccinyl-CoA in the presence of molecular oxygen. In other words, what we did here is we created our own enzyme for our own pathway. We're going to identify different enzymes, very different molecules. So we had enzymes in there for methylation. And this thioesterase would cleave molecule A into malate, this is shown here, and it would release the CoA residue, which could be fed back into the cycle. So in other words, by increasing complexity of the cycle, by adding an additional enzyme that would proofread and scavenge and recycle the stellar metabolite, we would be able to improve CO2 fixation efficiency. And in fact, what you can see, as soon as you add this thioesterase, you ramp up CO2 fixation efficiency. I think this is a very important principle, this metabolic proofreading, that we can use in synthetic biology. But I think it's also important to realize that in many biological systems, such metabolic proofreading principles are also operating. So that's one way, metabolic proofreading, to improve efficiency. Another way we can use is basically redesign our original strategy. And we have used this, for instance, to circumvent the reaction from propyl-CoA to this compound here called methylmalonyl-CoA. This was not a reaction that we could use because it made too many problems. So we had to go back and detour the whole reaction by going to a compound called acrylyl-CoA, and from there on then back to this compound methylmalonyl-CoA. The pathway redesign was very useful to us to identify enzymes that could do the job. And one other part of this design principle is, of course, to engineer enzymes to be more specific. And we've used